Hello, good morning and good afternoon, everyone. And I'm Zui Qian, Director of Marketing, Asia Pacific region with PacBio. And today I would like to welcome everyone to this edition of our PPAC webinar in collaboration with our partner in the region, Novogene AIT, with a focus on improving human health through genomics with Hifi, highly accurate long reads. I'm thrilled to present our guest speakers today, whom will present in the following order. Dr. Jonas Korlach, Chief Scientific Officer of PacBio, will talk about the latest technology updates in various sequencing applications using Hifi reads, followed by Dr. Emily Farrow, Director of Laboratory Operations in Genomic Medicine Center and Children's Mercy, Kansas City, who will share her experience on detecting pathogenic variants for rare and undiagnosed human genetic disease using Hifi reads, then followed by Ms. Tianran Shi, Senior Product Manager with Novogene AIT, who will introduce Novogene's PacBio sequencing service to empower researchers to harness the power of Hifi reads in human disease research. Before we get on with the presentations, I would like to quickly announce a few housekeeping rules. Due to the large number of audience calling in today, all listeners are muted. You can ask questions or make comments at any time during the webinar by typing into the questions box in the attendees dashboard. Please indicate which speaker your question is intended for. The presenters will then address your questions during the Q&A session following the presentations. With that, I would like to welcome our speaker again.
Hello, everyone. My name is Jonas Korlach. I'm the Chief Scientific Officer at PacBio. Today, it's my great pleasure to share with you a presentation entitled Hifi Sequencing, See What You've Been Missing. Hifi sequencing, or Hifi reads, are the cornerstone of PacBio's technology. On our Sequel IIe platform, you are able to access long, up to 15 to 20 kilobases long, and highly accurate, 99.9% accurate sequencing reads. This combination of long and highly accurate sequencing has made PacBio the gold standard for de novo genome assembly across species. For human applications, we're seeing significant adoption of Hifi sequencing for variant detection in translational research, either with a whole genome or targeted approach. And there's a ton of excitement for the use of Hifi in RNA sequencing. Hifi allows full-length isoform detection, and researchers are finding that either in bulk or at the single-cell level, this is providing a new window into biology that simply is not visible with short-read technology. And lastly, I'd like to mention epigenetics. In general, our understanding of the impact of epigenetic modifications on phenotypes of interest is in its infancy. There's been some fantastic work published earlier this year that is really starting to show the power of gathering epigenomic signals straight from the native sequencing run, getting methylation information along with the standard A's, T's, C's, and G's with no need for bisulfite conversion as an approach that will surely usher in a new wave of discovery. You may be familiar how Hifi sequencing reads work and what they are. In short, they're transforming genomics because they are 100 times longer than the average short read next generation sequencing read. For example, a 200 base pair Illumina read. PacBio Hifi reads can be up to 20,000 bases long, and this is drawn here on the left to scale. And they're as accurate as next generation sequencing reads. On the right, there's a graph showing the read accuracy distributions of the three major technologies. And you can see that PacBio Hifi reads are highly accurate, over 99% accurate, and very similar to Illumina short reads. And also that the distribution is very narrow, meaning all the reads are very accurate. And this narrow distribution makes the bioinformatic analysis much easier. In addition to the extraordinary accuracy and length of Hifi reads, they also provide other advantages, such as even coverage, high genome completeness, allele resolution, and long-range phasing. All these aspects taken together result in superior performance with respect to the calling of all variant classes. Indeed, both the recent Precision FDA Truth Challenge version 2, the Genome in the Bottle 0.6 benchmark, and included here are the most recent updates for the different technologies, highlighted PacBio Hifi data having the best performance and with substantially lower variant calling errors in SNPs, indels, and SVs compared to all other sequencing technologies. Touching on accuracy a bit more, I'd like to share a very detailed publication from the Association of Biomolecular Resource Facilities, or ABRF, that was recently published in Nature Biotechnology. The association compared all the different sequencing platforms, including the sequencing systems from Illumina, IonTorrent, PacBio, ONT, BGI-MGI, and Genapsys. The authors observed that PacBio had the lowest error rate of all technologies. In addition, PacBio Hifi had the highest mapping rate, lowest non-mapping rate, that is the highest proportion of useful data, and even in challenging regions like homopolymers and short tandem repeats, or STRs. Significantly, because of this superior accuracy, the authors found PacBio Hifi achieved the highest precision in calling clinically relevant variants compared to both short and other long-read technologies. Now let's put those long, highly accurate reads to work. Well, it all starts with a reference. What is the baseline that we compare things to? Many of us remember the publication of the first draft of the Human Reference Genome in 2001 and the quote-unquote completion of that reference in 2003. Tremendous accomplishments that have been the foundation now 20 years on for what now is called the field of human genetics and a driver of precision medicine. But as we all now know, the Human Reference Genome was not truly complete. It has taken the efforts of the Telomere to Telomere Consortium the last few years to finally and officially complete the human genome reference. The preprint for this work was published just a few months ago and marks a major milestone for the field. PacBio Hifi sequencing was essential for the completion of this work in that it provided the foundational technology from which the assembly was built and onto which data from other technologies were added. As the authors describe in this preprint, they leveraged the combined accuracy and length of Hifi reads to enable assembly of highly repetitive regions, even centromeres, and resolve closely related sequences, such as in segmental duplications. The work highlights the utility of Hifi reads to provide the most complete view of human genomes, finally resolving regions that have been impossible to see for many years. In fact, the authors summarized their findings, suggesting that at least 8% of the genome was previously overlooked and is now officially part of the reference, made accessible now with the long, highly accurate reads of Hifi. I would like to describe in this slide some of the benefits of utilizing Hifi reads for human genome sequencing, and do so by highlighting two additional consortia involved in human genome analysis work. The Human Genome Structure Variation Consortium, led by Charles Lee, Jan Korbel, and Evan Eichler, and the Genome in the Bottle Consortium, spearheaded by the National Institutes of Standards and Technology in the US. The HDSVC has shown that substantially more structural variants are resolved through PacBio sequencing, on the order of more than double of what can be derived from short-read sequencing. This was achieved from only 32 samples analyzed with PacBio compared to thousands of samples from the 1000 Genomes Project with short-read sequencing. In addition, on the bottom here, benchmarking performance on known and extremely well-studied samples, the Genome in the Bottle initiative found from only 3 samples analyzed with PacBio that substantially more single nucleotide variants and indels could be called in previously called quote-unquote difficult to map regions, and including 16% new exonic variants. So it is not just the structure variants that are being illuminated, but also SNVs and indels in what were considered difficult to sequence regions of the human genome. Beyond the completion of the human reference genome by the T2T consortium and the mapping of an additional 8% of the genome that was previously overlooked, PacBio is enabling discovery on multiple other fronts as well. First, I'd like to highlight the great work coming out of Dennis Lo's lab at the Chinese University of Hong Kong earlier this year. The team developed a methodology to directly examine 5-methylcytosine at the whole genome level using the kinetic information from real-time Hifi sequencing, without the need for bisulfite or enzymatic conversions. In this PNAS paper, Professor Lo et al. Described the development of algorithms that leverage Hifi sequencing information from both DNA strands to improve the detection of 5-MC. The paper describes its application to human whole genome sequencing and the benefits of characterizing the epigenome with accurate long reads. For example, allowing epigenetic phasing and allelic differential methylation. So just like the phasing of genetic variants, the ability to detect the methylation status genome-wide with long Hifi reads also allows for long-range epigenetic phasing information. Second, Trotsky et al. Out of the University of Queensland recently published a great example of the power of Hifi to resolve aspects of the human genome that were inaccessible before. The authors used deep, full-length PacBio cDNA sequencing of normal human tissues and cancer cell lines to hunt for pseudogenes. The authors identified many novel transcribed pseudogenes that were not in GenCode, and they further show that these are expressed in tissue-specific patterns. To assess the biological impact of non-coding pseudogenes, they CRISPR-Cas9 deleted a nucleus-enriched pseudogene and observed hundreds of perturbed genes. This study powerfully highlights pseudogenes as a complex and dynamic component of the human transcriptional landscape. And lastly, on this slide, I'd like to highlight a couple of exciting papers leveraging long-read RNA-seq or Iso-seq. For example, in this cool new preprint, Barbara Wolt and Ali Mortazavi used single-cell RNA-seq, single-cell Iso-seq, bulk Iso-seq, and ATAC-seq to make discoveries underlying the genomic basis of differential RNA isoform expression during differentiation, here from myoblasts to myotubes. They developed a new method called long-read split-seq, which allows for an understanding of how transcriptional start sites change through complex biological processes, such as cell differentiation. And in a second example for Iso-seq, this time in the area of cancer research, a group out of China recently published a paper establishing a single-cell survey of the cellular hierarchy in acute myeloid leukemia, again using full-length transcripts and cellular barcodes the different mutation profiles in these heterogeneous cancer types can be resolved fully with Hifi isoseq reads and assigned to the different cell types and clone lineages. But just as some researchers are using PacBio Hifi to push into the unknown, making new discoveries of basic biology and cellular processes, others are pushing on previous discoveries to bring them closer to the clinic. The first publication I'd like to highlight uses PacBio to resolve the variation in a human-specific tandem repeat that is associated with Lou Gehrig's disease, or ALS. Here, the authors used a targeted approach on PacBio to fully characterize the variation in length of the tandem repeat and its association to disease and its pattern of frequencies across human populations, further enhancing our knowledge of the role of the WDR7 locus plays in ALS etiology. Second, I'd like to highlight a recent paper about a Chinese infant with sudden cardiac failure. The researchers studying this patient had identified 2 mutations in the same gene with Sanger sequencing. However, it was not possible to determine whether these were in trans or cis. Using PacBio sequencing, which allowed the single molecule reads of over 8 kilobases, the researchers were able to determine that the 2 mutations were in trans. The first was inherited from the mother, and that the second was de novo on the paternal allele. The authors note that confirming the phasing of the mutations allowed clarification of the genetic etiology of the proband and offered benefits in patient management and counseling. And thirdly, another example of the power of long-range phasing is in the area of cancer research. As described in this science paper led by a team out of Memorial Sloan Cancer Center in New York City, researchers determined that multiple mutations in the PIK3CA gene are common in 12 to 15% of breast cancers, and of those, 95% are a double mutation. But these mutations can be very far apart and are difficult to phase with short-read sequencing or even Sanger sequencing. But with PacBio sequencing, they were able to resolve the phasing and show that patients with double mutations in cis show a phenotype of enhanced oncogenicity and greater sensitivity to PIK3CA inhibitors, to PI3K inhibitors. I've shared a few vignettes of how PacBio Hifi sequencing is making an impact in human genetics, but this is just the tip of the iceberg. Human genetics researchers are using Hifi as a new way of looking at human genomes, seeing regions for the first time or in a more complete context, and thereby allowing for new insights and discoveries in many areas. Additional area for impact as translational researchers bring this technology closer to the clinic include rare disease, pharmacogenomics, neurological diseases, cancer, reproductive health, or transplantation research, each with exciting opportunities and unmet needs that can be addressed with PacBio Hifi sequencing. Shifting to PacBio as an organization, we've had a very busy year, kicking off 2021 with the announcement of our collaboration with Invitae to accelerate long-read whole genome sequencing into routine clinical care, followed by a substantial investment by SoftBank into our company. We've announced a number of collaborations with world-leading organizations such as LabCorp, Children's Mercy Hospital, and Rady's Children's Hospital. And we've had a number of exciting product launches and acquisitions this year, namely Circulomics to extend our capabilities in extraction and sample prep, and Omniome for short-read sequencing technology. So with this, I'd like to thank you for your attention, and I'd like to leave with our mission statement, which is to enable the promise of genomics to better human health. And we do that by creating the world's most advanced sequencing technologies. We look forward to connecting with you to discuss how PacBio Hifi sequencing can assist in your research. Thank you very much.
Okay, well, I want to thank you for the opportunity to present our group's recent work at PacBio's ASHG workshop. Today, my name is Emily Farrow, and today I'll be discussing an integrated rare disease genomics using long-read genome sequencing. The Genomic Medicine Center at Children's Mercy Hospital was one of the first genome centers wholly embedded within a pediatric setting, and this was largely due to the recognition that rare disease is collectively not all that rare. In fact, there are over 7,700 genetic or inherited diseases. They affect 400 million people globally, which translates to 1 in 10 Americans, half of which are children. Molecular diagnostic yields by symptom-driven clinical exome sequencing in patients with rare disease only range from 30 to 40% in unbiased cohorts. And we now know that the addition of short-read genome sequencing only incrementally increases the diagnostic yield. One of the many advances in rare disease is the discovery of novel disorders and causative genes. In addition to the exponential increase in gene disease association, there is an increased recognition of the complexity of the human genome. Single nucleotide variants, while accurately and efficiently assessed with short-read genome and exome sequencing, are really only one component of molecular diagnostics. We now know that copy number variation, triplet repeat disorders such as Fragile X syndrome, methylation and imprinting disorders such as Prader-Willi or Angelman syndrome, large structural variations such as translocations that appear to be balanced but perhaps aren't, and mitochondrial variants are all contributing to genetic disease. So why are long reads increasingly being utilized and investigated in the context of rare disease? And that's because despite comprehensive sequencing using standard sequencing technologies and integrated informatic approaches, there are multiple remaining analyses challenges. While trio sequencing is powerful, parents are often not available for sequencing. And while this obviously limits the detection of de novo variants, which is critically important for neurodevelopmental diseases, it also is important for autosomal recessive disease, which still accounts for about 21% of cases. Even coverage remains a challenge, even in the face of PCR-free genome sequencing. Repetitive regions such as pseudogenes or expansion disorders, again, such as Fragile X, are difficult to align with short-read technology. And while there have been improvements in structural variant detection, there's still room for opportunities and improvement, particularly in the face of detecting inversions and translocations. And what this amounts to is that when faced with a patient with rare disease, we often are deploying multiple clinical evaluations with multiple assays. And collectively, these can be costly, they can be time-consuming, and they're limited to knowns, so we can only look at what we know. And in fact, an assay donut hole remains, where one technology can't detect all of the sequencing or copy number changes that the next technology would pick up. Historically, long-read sequencing has been difficult to deploy because of higher error rates and lower throughput. But as these have rapidly been improved, these are now an attractive solution to look at for rare disease. So in rare disease, when assessing for non-methylation, as I mentioned, it changes, laboratories are now utilizing a number of clinical testing methods, ranging from a karyotype to microarray sequencing, expansion, or even inversion testing. In some cases, differentiating which test is most appropriate is completely straightforward, such as suspected trisomy 21, you would start with a karyotype. However, in many cases, such as the case of neonatal hypotonia, testing algorithms are much more complicated. And again, what this amounts to is that with multiple testing assays, we have increased time and increased money. And if we look at average turnaround times for different technologies or assays that are used, we can have 4 weeks for a karyotype. You may have another 4 weeks for a microarray. Depending on which sequencing you're pursuing, it could be 8 to 12 weeks. If you're now suspecting an expansion disorder, such as myotonic dystrophy and hypotonia, that might be another 4 weeks, and so on and so forth. Now, the addition of copy number variant detection from short-read exome and genome sequencing does allow for some testing to begin to be condensed. However, again, these testing cannot detect expansion disorders. Informatic algorithms for PCR-free genome sequencing can screen for known expansions. However, these are still a screening mechanism that require orthogonal confirmation. Improvements in third-generation sequencing, such as the PacBio Hifi sequencing, offer the potential to capture complex copy number variants, single nucleotide variants, expansions, and inversions, simplifying our testing algorithms and offering the potential for an increase in diagnostic rates. So in recognition of the fact that over 50% of our patients who receive comprehensive sequencing, including exome sequencing, will not receive a diagnosis, the Children's Mercy Research Institute and the Genomic Medicine Center have created a novel research program called Genomic Answers for Kids. And under this program, we've created a pediatric data repository to facilitate the search for answers and novel treatments for pediatric genetic conditions. 30,000 children and their families will be enrolled over the next 7 years, and comprehensive sequencing and analytic strategies are deployed across the program. As part of our sequencing strategy currently, we now have a systematic reflex to Hifi genomes after a negative exome analysis has been completed. As you may imagine, to support the needed throughput for this program, we spent quite a bit of time optimizing our laboratory preparation methods and our analytical methods. As part of that, our DNA isolation has been completed using a Chemagic 360 automation, which allows for 24 to 48, all the way up to 96 samples to be isolated at once. Samples are sheared to 15,000 base pairs with an input of 5 micrograms of DNA. And library preparation is automated now with the Cyclone G3, which again allows for parallel processing of up to 96 samples at once. In our informatic or bioinformatic algorithms, we also have developed quite a few different strategies. Our sequencing alignment is completed using build 38 for all of our Hifi genomes. Our variant detection is completed using different algorithms, including deep variant, which is used for single nucleotide variants and insertions and deletions. WhatsApp is used for the phasing of the deep variant calls, and PBSV, or PacBio structural variants, is used to detect structural variation. For variant annotation, a novel toolset is used to annotate structural variants for population frequency and the impact on known genes. And our repeat expansions are also called tandem genotypes, are annotated for set loci. If we take a very high-level look at our QC metrics, what we can see with our Hifi genomes is that our single nucleotide variants and insertion deletion calls are highly concordant with short-read whole genome sequencing. In addition, the Hifi sequencing is revealing over 450,000 single nucleotide variants and insertions and deletions, and about 10,000 structural variants per genome that were previously undetected. If we apply a fairly conservative allele frequency filter of 1%, less than 1%, we now have about 150 rare variants per genome or per proband for our analysts to investigate that were not detected using our prior technologies. So while sequencing and analysis are ongoing, our initial analyses are already leading to novel discoveries and advancements, particularly in 3 challenging areas that I'd like to highlight today. An ongoing challenge that we have as analysts is the combined analysis of single nucleotide variants and copy number variants. In this case, CMH001017 and the siblings CMH001018 were referred for global developmental delay, hypotonia, strabismus, and MRIs that are consistent with a mild septo-optic dysplasia. And in fact, what was striking about these siblings is that they had a nearly identical clinical presentation. These families have a diagnostic journey of over 7 years that have elapsed since the first test has been ordered. And it's been quite comprehensive, with single gene analyses ordered, deletion and duplications of those single genes, a panel analysis, exome sequencing, and microarray, all of which were non-diagnostic. And then in this case, the family history was non-contributory, and we only have the 2 affected siblings for analysis. We completed Hifi sequencing for both the siblings and the parents. And what we found when this was completed was that they are compound heterozygous for 2 pathogenic variants and a gene AARS2. The first is a missense variant of C to T at position 595, which ultimately results in the replacement of an arginine with cysteine at codon 199, and this was maternally inherited. The second is actually a small deletion at 6p21.1, which includes the deletion of exons 5 through 7 in AARS2, and this was paternally inherited. Now, the deletion is a recurrent deletion that's actually previously been reported, but again, it's falling in this donut hole, whereby it's too small for conventional microarray, yet large enough to be challenging to detect with copy number from clinical exome. AARS2 is associated with a progressive leukoencephalopathy with ovarian failure, and in fact, the full symptoms of this syndrome fit quite nicely with our family. Another aspect that's been optimized is software solutions that are providing us visualization of the variants, allowing for efficient filtering strategies, pedigree analyses, and phenotypic-driven analyses. In this view, you can clearly see the loss of exons 5 through 7, denoted by the large red bar, with a single nucleotide variant with the small caret arrow that have only been seen in this family. Now, again, in this case, we had parental samples, but the power of phasing is evident as it is clear the deletion and pathogenic single nucleotide variant are in opposite alleles. So on the top, you can see the deletion of exons 5 through 7, and then on the right in the red bar, you can see on the second allele the single nucleotide variant. So again, this is consistent with autosomal recessive disease as the variants are in trans. So what were the challenges in this case? Two of them are that the deletions of exons 5 through 7 was not detected by microarray or exome sequencing. So again, it fell in that intermediate area that's difficult to detect. And the second challenge that's agnostic to technology, but an ongoing problem with rare disease, is that AARS2 related disease was actually reported in literature 2 years after the onset of symptoms in the proband and after the last clinical analyses, and not included even on the panel that was originally ordered. And so this again highlights the difficulties we have in keeping up with the new disease gene associations. In keeping with the theme of intermediate copy number changes that are difficult to detect, we had an affected duo that was also referred to our study due to multiple concerns, including growth deficiency, seizures, mild hearing loss, agenesis of the corpus callosum, mild white volume matter loss, severe scoliosis, global developmental delay, and dysmorphic features. And then in this case, the mother, CMH001061, had a very similar phenotype. The previous testing had identified a deletion at 16q24.3 that was not detected in the daughter when that array was completed. A comprehensive sequencing panel completed on the daughter, or CMH001060, was also negative. And after a negative exome analysis in our research program, we also completed Hifi genomes on the patient and her mother. And in fact, analysis of the Hifi genomes revealed that both the mom and the daughter carry the deletion at 16q24.3 that had not been reported by microarray in the child. So we do not have access to the original array data, so we cannot speculate on why the call was missed. However, this deletion encompasses exons 1 through 12 out of 13 in a gene ANKRD11, which is consistent with the diagnosis of KBG syndrome. And this syndrome is characterized by short stature, microcephaly, vertebral malformations, seizures in some patients, and developmental delay. The mother had had a diagnostic odyssey of over 30 years, and the probands had been over a decade. And again, this case highlights that intermediate deletions and duplications are still difficult to detect, particularly with older technologies with reduced resolution compared to the new third-generation sequencing. I think combine these two cases together really highlight, again, not only the difficulties in detecting these variants that we're now able to see with the deployment of Hifi genomes, but also the increasing recognition of small copy number variants and their role in genetic or rare disease. Building on our structural variant detection, another class of diseases are expansion disorders. Now, many classic genetic diseases are attributed to repeat expansion disorders, such as Fragile X, myotonic dystrophy, and Friedreich ataxia. Most commonly now, each test is completed independently using repeat primed PCR, replacing the traditional Southern blotting. And despite this huge advance, in many cases, the appropriate assay is still not completed, leading to the under-ascertainment of disease. And this is particularly true, we think, in Friedreich ataxia. Additionally, both the repeat number and the sequence and motif of the repeat are important. We now know that interruptions can modify disease severity in Fragile X, for example. So informatic algorithms for short reads are able to estimate expansions at known loci for PCR-free genomes, and they do actually well in their estimates. However, these algorithms don't give the full sequence or the interruptions. And so now in our program, each Hifi genome is analyzed for repeat expansions with a tandem genotype output that allows for quick scanning of repeat numbers. And then the expansion can also then be directly visualized using IGV. So in this case, CMH003372-04 was diagnosed clinically with Fragile X due to developmental delay. And this has been detected using the triplet prime PCR. However, the expansion number and the potential for sequence interruptions was unknown, as the report states fully expanded consistent with disease. So as a positive control to develop the tandem genotyping tool, a Hifi genome was completed, revealing an expansion of almost 200 repeats. As you can see in the top panel, the one allele, an average of the predominant allele is 200, which is consistent with the male with a single X and Fragile X disease. However, you also can see that there are some alleles with a larger expansion, which is not unexpected in a fully expanded allele with Fragile X disease. And in fact, when we look at within IGV closer, we can see that most of our alleles are having a 600 base pair, which would be a 200 repeat, triplet repeat expansion called. And we do in this view see a single expansion of 400 repeats, consistent again, both with our tandem genotyping tool and with what had been seen clinically. So we're particularly excited to deploy the tandem genotyping tool as expansion disorders are particularly common in neuromuscular disease, which is a common referral within our center. Again, currently, repeat expansions are graphed for 51 loci for each sample. And this has resulted in the carrier identification for Friedreich's ataxia and in females that have one allele consistent with Fragile X or expanded allele. Nicely, loci can be added and removed to the tool as the literature may report new expansions or as we've discovered novel expansions. And it's important to remember that the tandem genotyping tool gives us a quick way to screen for expansions, but the insertions are also called by the informatic pipeline, which has also allowed for the novel gene discovery of an expansion for a family we previously presented on STAR-D7. So although the sequencing of expansions is far improved with long reads, there are still some considerations and opportunities for development. Extreme expansions may be difficult to align even in the face of 12,000 base pair reads and may lead to reduced coverage to be aware of. Some specific repeat motifs are difficult to sequence, such as GAA for Friedreich ataxia. And in this case, what we can do is we can do a manual inspection of all the reads, and that can be very helpful in resolving what the expansion and what the repeat insertion is. And we've been quite successful in this regard. Mitochondrial disease is another often underappreciated disease mechanism. It affects nearly 1 in 5,000 births, and onset ranges from more severe presentations in childhood to milder presentations in adulthood. Symptoms are wide-ranging and can include myopathy, lactic acidosis, seizures, and ataxia in childhood, to again milder presentations such as hearing loss and migraines in adults. The mitochondrial genome itself is only 16,569 base pairs with 37 genes. In contrast, the nuclear genome encodes over 1,000 mitochondrial genes important for mitochondrial function. And despite the difference, pathogenic variants in the mitochondrial genome, including a recurrent 5,000 base pair deletion, account for just a little under 15% of mitochondrial-related disease. Therefore, particularly in rare disease, evaluating the mitochondrial genome is important. With sample preparation modifications in our hands, the Hifi genome now captures the mitochondrial genome with an average coverage of 100 to 200x per sample. So again, using the current automated sample preparation techniques that we have, we've preserved the mitochondrial genome, allowing for a complete analysis. And in some cases, the entire mitochondrial genome is covered in a single Hifi read, providing the ability for accurate copy number variant detection, again, a common disease mechanism. Further, our integrated software solutions also include the analysis and visualization of the mitochondrial genome. The one caveat is that in some cases, low level of heteroplasmy, which would look like a low variant allele fraction, in this case would not be detected if it's below 1% or very low in our samples. So if we look at the progress we've made with our Hifi genomes, we've now completed over 600 Hifi genomes to date and still counting. Again, the sequencing and analyses are ongoing. And in our diagnostic findings, we found multiple copy number variant deletions and duplications that are attributed to disease. We've discovered single nucleotide variants, and we've also found repeat expansion disorders that were not found previously. The only thing is we're essentially missing methylation currently with our techniques, which is still important for imprinting disorders, and at this point would still require reflex testing. However, we've shown that what now may take 6 tests can potentially be done with 2 tests, which is a significant improvement. So in summary, what we've shown is that we've been able to automate the sample preparation to support the large-scale deployment of Hifi sequencing in a rare disease program. The comprehensive integrated analyses allow for the simultaneous detection of multiple types of genomic variation, providing cost and time savings. And ultimately, the systematic implementation of Hifi genomes continues to lead to new diagnoses previously unidentified with standard technologies. And I would really like to take a moment to acknowledge our team, particularly our director, Dr. Tomi Pastinen, and all the coworkers, both within Children's Mercy and within PacBio that we've worked with in collaboration. And most importantly, I'd really like to acknowledge the patients and their families for their participation in this report, because without them, we wouldn't have a program. And with that, I thank you for your time.
The Genomic Medicine Center at Children's Mercy Hospital is pleased to offer an NHGRI-sponsored R25 short course, June 6th through 9th, 2022. This 4-day immersive course will be offered with virtual and in-person options. The course covers technical aspects of next-generation sequencing, variant interpretation, and includes hands-on guided analysis workshops. Please see our course website for additional details.
Hi, I'm Tianran Shi, Senior Product Manager of Novogene AMEA. I'm so happy to be here in this webinar together with Emily and Jonas to share what's in store for long read sequencing. And I'm going to share how Novogene PacBio Hifi services can empower you in your human disease research. And next, I would like to take some time to introduce Novogene to you briefly. Novogene is the global leader in sequencing services. Founded in 2011 and headquartered in Beijing, China, our mission is to use advancing genomics to improve human life and health. And we have global presence and labs across the continent. We are located in Singapore, China, US, and UK. Our AMEA regional headquarters is located in Singapore, offering genomic services to customers in Asia Pacific, Middle East, and Africa, as well as across the region through our network of local partners. And our Novogene AIT lab includes single-cell lab, and we will bring you single-cell services from Japan this December. And there's a great news recently. Novogene enhances its long-read sequencing capacity with the addition of more than 10 units of PacBio Sequel 2 and Sequel 2E system. And now we have more than 10 PacBio Sequel system in China. And we also have new PacBio Sequel 2E in Novogene AIT sequencing center in Singapore. And why should you use Novogene services? Firstly, we have dedicated professional technical support, and they have rich experience in solving various problems. And we developed and launched our real-time customer service system this year. In this CSS system, you can track and view project status, submit sample information, and obtain sample QC and data analysis report, and also download your data. And helping our customers to achieve their research goal is what truly matters. That is why we place the utmost importance on our services, on providing reliable services with extensive experience and shorter turnaround time. Here shows our achievements over the years. Our sequence services have allowed scientists to publish tons of papers in top journals like research organizations and universities, hospitals, and companies. And here are Novogene end-to-end solutions. Our services include genome sequencing like human, plant, animal, whole genome sequencing, human, plant, animal de novo sequencing, and whole exome sequencing. We also have transcriptome sequencing like mRNA sequencing, non-coding RNA sequencing, isoform sequencing, and also single-cell gene expression sequencing, spatial transcriptomics sequencing. And we also have microbial sequencing like shotgun metagenomics, amplicon metagenomics, and 4-16 metagenomics, and also have microbial de novo sequencing and resequencing. We have epigenomics like ChIP-seq, RIP-seq, WGBS, and we also have oncology services like NovoPM 2.0, NovoFocus AR, and clinical WES. Today, our topic is focusing on what kind of services can we offer using PacBio to help your human disease research. Today we'll go through 4 parts. The first part is the Novogene PacBio service overview and workflow. The second part is the introduction of PacBio Hi-Fi services. And the third part is application of PacBio Hi-Fi services for human disease. And the last part is one case study. And firstly, this is the overview of our all PacBio services. So we can offer de novo genome sequencing for species without reference genome, and human whole genome sequencing, plant animal whole genome sequencing. The microbial genome sequencing include full-length amplicon sequencing, metagenomic sequencing, and de novo fungal and bacterial sequencing. We also have the full-length transcriptome sequencing, also called isoform sequencing. As we all know, Novogene has a strict quality control workflow. So once your sample is ready, we will do sample quality control. So we will use gel electrophoresis to test your purity and the integrity of your DNA to make sure your sample is clean and didn't degrade. And the PFGE is used to test if the DNA size is qualified. And we will also use the QB to get the concentration. As long as your sample passed our QC testing, we will perform library preparation using PacBio library prep kit. Then we will do library QC to make sure it could be sequenced on PacBio systems. After we get the raw data, I think the data QC is necessary before doing bioinformatic analysis. Next, come with me to learn more about our PacBio Hi-Fi services details. The human whole genome sequencing with PacBio high accurate long reads can generate complete and fast human genome assembly of discovering populations to better understand the complexity of health and disease. So we recommend at least 20-fold sequence depth with 15 kilobase pair insert size and using the CCS mode to get your Hi-Fi reads. And here are our standard analysis contents. We will do structure variation and copy number variation calling annotation after the alignment with the reference genome. These 2 tables from our report show the statistics of SV and CV variation detection and annotation results. Copy number variation refers to the increase or reduction of copy number of large fragments in the genome. Abnormal CNVs change can be linked to various diseases. The gain here means the duplication and loss means the deletion. This table shows the number of CNVs in various genome regions. And the structure variation refers to large structural variation in genome, such as deletion, insertion, inversion, and duplication, and also translocation. Copy number CNV, SV is prevalent in human genome and can be source of individual difference and the disease susceptibility. Next service is the full-length transcriptome sequencing, also called isoform sequencing. The picture workflow left on the left shows our analysis workflow, and IsoSeq we all know is an ultra high throughput method for discovering new genes, transcripts, and alternative splicing events. It can also improve genome annotation to identify gene structure, regulatory elements, and coding regions. So combined with the NGS RNA data, it can also increase the accuracy of RNA quantification with isoform level resolution. The next sequence is the full-length 16S emptycan metagenomics. It is frequently used to profile complex microbial community composition at the species or strain level. However, the short reads produced by NGS cannot provide taxonomic classification below the genus level. This panel depicts the unclassified percentage of species of variable regions. So only the full-length sequencing V1 to V9 can provide unbiased resolution of all species. So what are the main advantages of current long-read sequencing in medical genetics? The predominant difference between long-read sequencing and the short-read sequencing is the significant increase the read length. In contrast to the short-read, long-read sequencing has the capacity to sequence on average over 10 kilobase in one single read. So it requires less reads to cover the same genes. So aside from reducing alignment and mapping errors, long-read sequencing holds various advantages over short reads. So firstly, it can improve the detection and characterization of large structure variation, like large inversions or translocations. And the second one is it can sequence over long tandem repeat expansion and extreme JC high regions. And it can also enhance phasing or improve the discrimination of clinically relevant genes from their pseudogenes. This list shows the human genetic disease investigated with smart sequencing. Like we just said, is 4 parts. The first part is structural variation, and the second part is the tandem repeat expansion. Tandem repeat is a region of genomic DNA with multiple copies of short sequence units, like 1 to 6 base pair. These repeat regions are highly mutable due to the replication errors that occur during cell divisions. And to date, over 30 human diseases known to be caused by tandem repeat expansion, like Fragile X and the Parkinson's disease. For the phasing refers to the means of assigning genetic variants to the homologous paternal and maternal chromosome. In medical genetics, phasing is very important for understanding inheritance patterns, and the allele-specific expression. The last one is the pseudogenes. Pseudogenes usually originate from gene duplication or retrotransposition events and are defined as sequences that resemble known functional genes but cannot produce functional proteins. Next, we will go to the case study. This case study today is the long-read genome sequencing identifies causal structural variation in Mendelian disease. As we all know, short-read sequencing has limited ability to evaluate repetitive regions and structural variants. But long-read sequencing has the complementary strengths, and this study aimed to determine whether long-read sequencing could offer a means to identify overlooked genetic variation in patients undiagnosed by short-read sequencing. So they performed the low-coverage genome long read sequencing to identify structural variants in a patient, an Asian Hispanic male, and who presented with multiple neoplasia and cardiac myxomata, in whom the results of targeted clinical testing and genome short-read sequencing were negative. The long-read sequencing approach yielded 6,971 deletions and 6,821 insertions over 50 base pair. And after filtering for variants that are absent in an unrelated control and overlap a disease gene coding exome, identified 3 deletions and 3 insertions. One of these is the heterozygous 2,184 base pair deletions that are included in the first coding exon of PRKAR1A, and 2 of 4 reads at the local support this deletion. And they also use the Sanger sequencing and the Illumina short read sequencing to support the deletions. As you can see, the Sanger sequencing results confirms the deletions. The forward 1073 and the reverse 1074 from a representative applicant agree to the base pair with the higher quality of PacBio read. And also the Illumina short read supports the heterologous deletions variant through a drop in read coverage and clipped reads at the deletion breakpoints. So short read sequencing has decreased sensitivity for insertion and deletion variation detection as the size of the event increase, and it can miss up to 80% of structural variants in an individual genome. So this case demonstrates that the ability of genome long read sequencing to effect the detection of chiral structural variation in a rare disease. By the way, Novogene presents you with the new PacBio Sequel2E system. So this exciting platform for long read sequencing that delivers highly accurate long read. We're rolling out an exclusive promotion that can offer researchers the opportunity to get on a journey of applying long read lengths and high accurate sequencing for their research. So for more information about our promotion, you can scan the QR code to learn more. Or you can contact our local commercial team, and they will be very happy to help you with your long read sequencing journey. And that's all. Thank you.
Okay. Thank you, all the presenters, for your excellent presentation. I hope the audience, you enjoy the 3 talks as much as I do. So now we're getting to the Q&A session. So looks like we got quite a number of questions already coming in. So let me start over from the very top. Just to remind everyone that if you have any questions, comments, please type them into the questions box shown on the dashboard. Okay, so let's look at the first question. This is for Dr. Farrow. From your experience, what were the main barriers for the adoption of a Hifi sequencing in Children's Mercy Hospital? And what would be your advice for other hospitals looking into adopting whole genome sequencing using Hifi?
So thank you again for allowing me to participate. When we were getting started, I think the biggest barrier for us is that we didn't have any of the sequencers on site, and we hadn't had the chance to really work with anybody that had a lot of experience with the long reads. So we just weren't familiar, I think, with the system. And so I think now a lot of those barriers already have changed with the addition of the Sequel2E, and there are a lot more resources and a lot more of us that are using it. So I think as an advice, in addition to having to plan for the capital acquisition, which is always going to be something I think about, I think the best thing to do would be to talk to people who are already using
the sequencers or have experience with the data outside of PacBio, so at other institutions, to see how they're using it, how it's been successful. And we've been quite successful too in being able to adapt the library to our existing workflow so that we haven't had to come up with different streams for different sequencers. And that's been really important for us. And once we got that figured out, it was kind of like a magic switch as well.
Excellent. Thank you for that insight. Next question, I guess, is also designed for you, Dr. Farrow. Looks like in your experience so far, is cost still a concern for whole genome sequencing for rare disease diagnostics? This question is with regards to both the whole genome sequencing in general as well as specifically for Hifi.
Yeah, so I think unfortunately cost is something that we definitely consider, particularly on the clinical side, for the way our billing structure is, because our exomes in general, if we're honest, have gotten much, much better. And so we can get a lot of our diagnoses with a really pretty cheap and really efficient exome. And we've even gotten much, much better at our copy number from exome. And so that's where we really started to look at then if we have our negative exomes, where should we spend the money on our cost? And that's where we've now decided on our research side that we are deploying long read sequencing because we're getting much more out of our genomes by long read sequencing. And we're really only getting kind of small incremental gains with a short read genome. So that's where we've decided the best way to utilize our resources are.
Thank you for that. Let's see. Next question looks like it's for Dr. Korlach. So in your presentation, you actually presented one case. This is on the methylation study done by Dennis Lowson. So can you comment on the difference between that particular study versus what was actually presented by Aaron Wenger of PacBio during the ASHG conference recently?
Yeah, thank you. Certainly, yeah. So during the ASHG conference, which just happened, my colleague Aaron Wenger presented the latest work. Both methods are conceptually similar, but we've done some more benchmarking and training of the models, and we've built some tools that are streamlined and the bioinformatic analysis with regard to the data files. And this is now available for early access for collaborators who have, advanced bioinformatic capabilities. We're certainly developing this further. And so there's some differences there, but overall, the two approaches are conceptually similar.
Great, thank you. Next question regarding the circulomics acquisition that you mentioned during your talk. So can you tell us a little bit more about the circulomics product offering in the context of PacBio Long Reads Sequencing?
Yeah, certainly, I'd be happy to. And so the overall strategy is that we realize that an overall genomics program is not just a sequencing system and generating sequence data. It goes all the way from extraction of the DNA all the way to the data analysis and then the interpretation. And we want to be a partner with the scientists such as Dr. Farrow and with the entire scientific community to help and assist as best as we can in all of those areas. Specifically, making high quality DNA upfront is critical to get good Hifi sequencing, generate the best possible data. And so we wanted to invest in that area, and the company Circulomics has developed and products and expertise and a lot of experience over the last 10 years. And so it was for us a really natural fit to bring that expertise and acquire the company to bring this in-house. And it's been a great experience since we acquired we've immediately connected. Obviously, we worked with them already before, so it was a seamless transition. And we're already seeing now the application of the Circulomics products, the nanobind technology that has been around for a while, now even more focused and with more direct support from PacBio, applying either their products or, frankly, if there are other products that work for particular tissues and particular questions, we just want to find the best solution for the customers to get the best DNA quality going into the sequencing machine.
Excellent, thanks. Yeah, I know that a lot of the customers have been crying out loud, for a very robust DNA isolation protocol specifically for PacBio Long Read technologies. Now, finally, this is something come to fruition. Yeah, it's great. It's going to be great for the scientific community in general, I think. So the next question, I think, is for Tianran. So what is the analysis software used for the full-length 16S analysis? Is it KIME1 or KIME2?
We use the CHAM2 for the full-length 16S amplicon analysis. And the difference between the CHAM1 and CHAM2, the CHAM2 is the upgrade analysis pipeline for amplicon analysis, and it can do the taxonomy annotation on the real sequence. So the species taxonomy annotation resolution will be higher than the CHAM1 analysis pipeline.
I see. Looks like the next question is also for you, Tianran. What is the format for data release in this promotion that you will describe?
In our promotion, the format of our data release is only we only offer Hifi data release.
I see.
Hifi reads is our data release format. Okay.
Thank you. The next question is for Dr. Farrow. Are you building up a healthy control long read SV database besides using the publicly available ones?
So what we're doing is whenever we sequence a proband, we always are sequencing the parents. So we're prioritizing trios. Generally, those parents are healthy, and we are working to release all of our data, or at least make it accessible to everyone in the research community. So we recently released a paper on bioarchives, and that explains how to access that data. That in addition, you'll be able to see the phenotype for the proband. So it's not a pure control in that it is a family member of someone that has a rare disease, but in general, the parents are healthy. And so that's our strategy for getting more controls. And we absolutely recognize, along with many other leaders in the field, that we need to have better structural variant databases to allow people and really leverage all of the data that's being generated.
Thank you. Next question is for Dr. Korlach. How much improvement can be achieved in the diagnostic yield using Hifi reads compared to, let's say, WES?
Yeah, thank you. There are a number of research studies, but I would say by far the most extensive and most informative is the study by Dr. Farrow and her colleagues at Children's Mercy Kansas City, and the preprint that she just mentioned, which was posted in the Med Archives not too long ago. I'm going to post to all of you right now the link to that preprint if you want to check it out. It's really a terrific study looking at over 1,000 human alleles and showing that the rate of explaining the underlying cause of the disease is increased in this particular cohort by 13% by including structure variant information. And the paper gives a very nice and detailed explanation for why this is and what additional benefits the Hifi data provide. A doubling of the structure variants, a fourfold increase of the rare coding structure variants, and the phasing of the even the SNPs. So there's a 5% increase in the single nucleotide variant discovery rate, and that may not sound like much, but 5% SNPs is about 200,000 additional single nucleotide variants that you can now see and phase to their haplotype. So it's a beautiful study, so in this particular study was 13%. I would note, however, that we can expect the diagnostic rate to vary relative to the type of disease, the type of cohort, and so forth. Let me give you an example. In some cases, you can tell with short read sequencing that there are 2 mutations in the same gene. but you cannot phase them. You cannot understand whether those two mutations are on the same chromosome on cis or in trans on the two different chromosomes. And Hifi sequencing essentially has 100% diagnostic rate for this type of question, because in almost all cases we can span those distances and you can understand the phasing. And then so that's like 100% for those particular cases. And then on the other end of the spectrum, there may be cases where it's not even clear whether it's genetic to begin with. And so there the diagnostic or the solve rate would likely be quite a bit lower. So, you know, I would expect in areas, for example, neurological diseases and repeat expansions, which Hifi sequencing can resolve really well, that the additional explanation rate would be quite substantial. And so it can vary, but what we've seen from the researchers at Children's Mercy, also other studies like at Hudson Alpha Institute for Biotechnology and other sites, it is significant, and really from what we're seeing, it justifies the application of Hifi read sequencing to really give you know, the researchers the tools to understand more about the biology of these cases.
Thank you for that elaborate answer, Jonas. The next question, I think it's directed for Dr. Farrow. While the diagnostics is important, do you foresee them leading to any actionable target?
So we actually are already seeing where we can use Hifi and we need that for treatment. So one area is a kind of new area under development is our Aso Targeted Therapies. And so for these and rare disease, we're targeting a specific variant, a diagnostic variant, but what we need for that targeting and for it to work is that we need to have accurate haplotype phasing across the genome. And the only way we can do that reliably and to get the phase haplotype blocks that they need is if we use Hifi sequencing. And so we have now a new collaboration where we're working with NLORM to help them with some of their patients to provide the sequencing so that they can develop the therapies. And so that's just one that's already in practice and is already needed. I anticipate that there will be even more recognition where we can, the extra information that we're getting from the long reads will be critical for our precision guided therapeutics.
Yeah, because the antisense oligos, you're targeting the deleterious allele, and you need to specifically target that by leaving the wild type allele alone, right? And so that's why the phasing information is so critical.
Yeah, awesome. Yeah, thank you. So Jonas, next question is for you. Can we carry out the multiplex of libraries in the single smart cell for Hifi sequencing?
Yes, absolutely. So that's fully enabled, and we have barcodes. There are different strategies. So for whole genome sequencing, most often barcoded adapters are used during the library preparation, and then the samples can be pooled. And for targeted sequencing, we have multiple different options, having barcodes either in the primers or in a two-step PCR, where you first have a specific primer for the first step PCR with universal linker, and then you barcode in the second round of PCR. And the smart link and smart analysis software is fully enabled to then demultiplex those samples and give you the answers for each particular barcode. So this is now very mature.
Thank you. I think I should close up pretty soon because we're already 15 minutes over the time. So last question. This is for Jonas. Perhaps Dr. Farrow, you could also chime in. The question is asking about the accuracy between Sanger and Hifi from PacBio. Which one is better? Give your perspective. Yeah.
So maybe I'll start. So there is actually a paper that was published in 2018 by a research group in Canada, and the title of the paper is A Sequel to Sanger, Amplicon Sequencing That Scales, directly comparing the accuracy of Hifi reads and the Sanger sequencing technology, and they found that the Hifi reads are more accurate, especially in areas that are challenging for Sanger sequencing, like homonucleotide stretches or extreme sort of GC-rich regions and so forth. And so The accuracy distributions that I showed are for all different sequence contexts, Q30 plus reads. And so what we and others have found that the accuracy is on par for regions that are easy to sequence with both technologies, with Sanger technologies, and then the Hifi reads excel in regions where the Sanger sequencing technology has difficulties. And then obviously, in many cases, you have challenges because the Sanger sequencing is sequencing both alleles. And so when you have heterozygous SNPs, you have both peaks and so forth. Because Hifi sequencing is a single molecule sequencing technique, the alleles are separated in independent sequencing reads, which is an advantage. In addition, as you know, in Hifi sequencing, both the forward and the reverse strand are being sequenced of that same DNA molecule, and that gives you additional confidence because the sequence context, of course, of those two strands are different, and then you build consensus from that same double-stranded molecule.
Emily, over to you.
Yeah, so I would agree. So the nature of our study, because it's research, when we need to return those back, we have to do a clinical test, and that clinical test is 99% of the time is a Sanger confirmation. And so again, in areas that are relatively easy to sequence, they perform similarly. I think we still recognize that every sequencing modality has its own error type, and so as we've gotten more familiar with Hifi, we can find a few of those signatures, and sometimes, you know, Sanger can help us with those, but often there's different regions. So I would agree with everything that Jonas is saying, and that they're both great techniques, but we're obviously finding the variance through Hifi, and the Sanger is a mechanism for us to report it back.
Thank you. I apologize for the ambient noise I'm getting here. So again, thank you for all the presenters. Thank you for your excellent presentation, and thanks everybody for attending this webinar. We're actually going to make the recording available to you later, so watch out for the another email announcement with the link to the recording. So once again, thank you everyone. Have a lovely day. Bye bye.
Thank you very much. Bye bye.
Thank you.
Thank you.
Thank you, everyone. Bye bye.